few words about Andreas. Andreas is one of the leading scientists in drug delivery, in particular in transmucosal drug delivery. I always say that Andreas is the #1 scientist in the area of mucoadhesion who pioneered the Thiomer polymers, and he will be speaking about Thiomers today. Andreas published more than 500 papers and also received numerous awards. Okay. Yeah, we can see your slides now.
Okay, so can you see the presentation?
Yeah, we can.
Perfect. I will also activate the laser pointer. Yeah, Vitaly, thank you very much for inviting me. Within the next 45 minutes, I would like to draw your attention to the biomedical applications of Thiomers, and in particular, I will focus in my talk on new developments, clinical trials, and also on advanced products. Thiolated polymers, designated Thiomers, are simply polymers bearing thiol groups. We started with the design of such polymers already more than 20 years ago, with the intention to improve the mucoadhesive properties of, in those times, well-established polymers such as polyacrylates or alginates or chitosan. And we did so being aware of that within the mucus gel layer, we find cysteine-rich subdomains of mucins. And our working hypothesis was, when we attach thiol groups on well-established mucoadhesive polymers, will then these thiolated polymers form new disulfide bonds within the mucus gel layer. That was our working hypothesis, and that gave more or less birth to the development of thiolated polymers. We started with polymers such as thiolated polyacrylates, thiolated chitosans, and also certain thiolated cellulose derivatives. After these first developments, and we filed the first patent on thiolated polymers in 1998, more and more research groups moved in those times new fields, and we all, the whole community working on thiolated polymers, realized that these polymers do not just show improved mucoadhesive properties, they show also a lot of other interesting properties, not just for drug delivery, for which high mucoadhesive properties are highly advantageous, but also potential for the design of various medical devices, potential in the field of regenerative medicine, and also potential for other fields such as cosmetic applications. You can attach thiolated polymers via the formation of disulfide bonds also to cysteine-rich structures within the keratin we find in here, also potential in textile industry, etc., etc. You might get a quite good idea about the potential of thiolated polymers for life sciences by the simple imagination of protein chemistry with and without cysteine. Having said that, of course, you can then say, well, why aren't they using directly proteins? And the reason for that is illustrated here. Proteins are simply too complex. When we use them in vivo, they bear a quite high immunogenic character. We know that they are unstable. They show a lot of additional functions we in principle do not need, or they are even disadvantages. And quite often the degree of thiolation is too low for the purpose for that we want to use these systems. But of course, when we use polymers like polysaccharides to which we attach thiol groups, we more or less mimic the workhorses of our body. We mimic the function of proteins. In the late 1990s, we were proud that also other research groups followed our concept. So we started counting publications on thiolated polymers coming from other research groups. The number was strongly increasing, so we stopped counting that. We then started counting clinical trials on thiolated polymers, and meanwhile there are I think almost 30 clinical trials on thiolated polymers available. So we also stopped counting, I think last year, the number of clinical trials on Thiomers. We then counted also products on the global market containing thiolated polymers, and we have also stopped counting that. So it's really now booming the entire technology. But before I will go in detail on certain product developments, let's go back to the basics. Let's go back to the synthesis of thiolated polymers. So that's a quite busy slide. I won't go too much in detail on that. We reviewed actually a couple of years ago the different techniques you can use to attach thiol groups on well-established polymers such as polysaccharides, polyacrylates, etc. The perhaps only reaction I want to explain to you in more detail is shown not on this slide, it's shown on the next slide. This is a substitution reaction we started to use quite recently, and that's a method that I can really recommend. Here we thiolated cyclodextrins, but the method works also for polysaccharides. In principle, you need just hydroxyl groups, and these hydroxyl groups can be quite easily substituted by thiol groups under the use of phosphorus pentasulfide, and that's really an efficient reaction. It is illustrated right here. Within a few hours, you can entirely substitute all hydroxyl groups by sulfhydryl groups. To keep a long story short, the thiolation of well-established polymers is quite simple. You find numerous techniques to do this thiolation. Meanwhile, the scale up for certain thiolated polymers has already been done, and you find also for certain thiolated polymers GMP materials, so that you can go also in the clinic and do clinical trials with these polymers. Why are we thiolating polymers? The reason for that is that as already explained to you in my introduction, we can really tremendously improve certain properties of well-established polymers, not just the bioadhesive or mucoadhesive properties. We can also improve various other, I would say, key properties for biomedical applications. And within the rest of my talk, I will go in more detail on these key properties and why they are the advantages for various product developments. Having already started with the improved mucoadhesive or bioadhesive properties, I would like to continue first of all with that. I mentioned in my introduction that, and that was our concept when we started with the design of this new type of polymers, that based on, for instance, an oxidation process, you can form new disulfide bonds, but also based on thiol disulfide exchange reactions, you also form new disulfide bonds. And the reason for this tremendously improved mucoadhesive properties is even more complex. You have to keep in mind that these polymers also penetrate into the mucous layer, so-called interpenetration process. Then they can even form new disulfide bonds within their own chains. And in this way, they get additionally anchored within the mucous gel layer. So how do these highly mucoadhesive systems perform in vivo? I want to show you just a few examples on that here on a thiolated chitosan. These studies were done in rabbits, and when you use unmodified chitosan, even after 1 hour, most of your polymer is already eliminated from the ocular surface, whereas when you use the thiolated version of chitosan, even after here 22 hours, you still find a significant amount of dethiolated polymer on the ocular surface. Here are results of clinical studies. We designed a thiolated polyacrylate and compared it with the unthiolated polymer using fluorescein as marker. And when you compress mini tablets inserts for the administration of these devices, they swell then in the tear fluid. They look actually like contact lenses. You see them here in the eye, and they are highly adhesive, and they remain there, as you can see here, at least for 8 hours, providing a sustained release of the incorporated drug. Still, these inserts are to some extent irritating, and when you go for simple gel formulations, they might also be irritating, and also your vision might not be anymore provided in the way it should be. And that gave actually birth to thiolated cyclodextrins, which we call the invisible choice to prolong ocular residence time. Here are data on in vitro studies where you can simply see the more thiol groups you immobilize on cyclodextrins, the longer is the mucosal residence time. in collaboration with an Italian research group, we went then in vivo in rabbits, incorporating in thiolated cyclodextrins dexamethasone. And as you can see right here, these are the red bar is the relevant bar when you use the thiolated version of cyclodextrin, you can really tremendously prolong the ocular residence time of this therapeutic agent. And I won't go in detail, but on the right-hand side, these images provide evidence that these thiolated cyclodextrins are also well tolerated by rabbits. Coming to another mucosal tissue, the intraoral mucosa. Here I would like to share with you results also from clinical trials with the thiolated alginate. And in this study, in the thiolated alginate film, metformin was incorporated, and the residence time in the intraoral cavity was also tested. And what could be shown was that when you use the unmodified alginate, these films are displaced within the first hour, whereas when you use the thiolated polymer, it takes much longer. In fact, even after 12 hours, still a significant amount of this thiolated polymer was still on the intraoral mucosa. This is another study providing also evidence in humans for the prolonged residence time here of thiolated nanofibers. Most challenging regarding a prolonged residence time is certainly the gastrointestinal mucosa. The reason for that is that we have a quite rapid mucus turnover. So when you anchor your polymer just on the surface of the mucus layer, even if it is really properly anchored there, you won't really see a prolonged residence time because of this mucus turnover process. And it took us quite a long time to achieve also a prolonged gastrointestinal residence time, although this is highly demanded by the pharmaceutical industry. And the reason for that failure was that when you use polymers with long polymer chains, they do not sufficiently penetrate into deeper mucus regions, and then they are subject to this mucus turnover and they are quite rapidly eliminated. But when you use thiolated cyclodextrins, they are small enough, they can really nicely penetrate into the deep, firm mucus layer, get anchored there by the formation of disulfide bonds. And finally, we could show also a really tremendously prolonged gastrointestinal residence time of this type of thiolated polymers, namely of thiolated cyclodextrins, which is illustrated here. Apart from this improved adhesive, mucoadhesive properties, thiolated polymers show also strong in situ gelling properties, and this is illustrated here. It is likely the most important application for these in situ gelling properties are products in the field of wound healing, in the field of tissue engineering. As you can see right here, you can inject thiolated polymers in liquid form. And then when they have been injected, they oxidize, so they form disulfide bonds. The viscosity is strongly increasing, and in addition, cells that are immigrating in these scaffolds, they bind also to thiol groups we find on the thiolated polymers. So that's then not mucoadhesion, that is then a bioadhesion process that is also taking place. Apart from a cross-linking via the formation of new disulfide bonds via an oxidation process, also thiol disulfide exchange reactions are involved. And you can also make use of thiol-in reactions to cross-link thiolated polymers. We went in detail on all these different types of cross-linking mechanisms in a review article I can recommend to you. There we really describe the chemistry behind how such cross-linking processes can be nicely controlled and tuned. Here you can see the results of the increase in viscosity of a thiolated chitosan. And we could show in this study that even within 5 minutes, you see an increase in viscosity based on this formation of new disulfide bonds of even 10,000-fold. So you have first the liquid, as I said, and after the oxidation, within 5 minutes, it's really a very stable three-dimensional network that you can use also for tissue engineering. Also, research groups, they made, of course, the same observations. This is done from a study that has been performed by a Chinese research group also with a thiolated chitosan, also showing this tremendous increase in viscosity, and that you get then really very stable three-dimensional scaffolds that can be used also for wound healing or tissue engineering in general. Also nice, I just wanted to add also at least one slide in my presentation on the thiol-in reactions. And here, this is also a study that has been done by another research group. They could show that you can really nicely cross-link thiolated cyclodextrins with this the allyl pack short-chain polymers, and they get really crystal clear gels, as illustrated here by making use of these thiolated cyclodextrins cross-linked via these thiol-in reactions. Results from clinical trials, I added here just one slide on that, focusing on the re-epithelialization of corneal wounds. I'm sorry, it's a busy slide. I won't go in detail on all these results. Perhaps just go in detail on the findings. What they could show is that the thiolated carboxymethyl hyaluronic acid they used in this study showed a high ability to accelerate corneal wound closure even within a day, and they also reported that this thiolated was safe within this clinical trial. And meanwhile, there are quite a lot of clinical trials going on, focusing on these wound healing properties of thiolated polymers. When you combine then the mucoadhesive or bioadhesive properties and the in situ gelling properties, so when we discuss these two properties jointly, then there are also products, making use of both of these properties at the same time. I think a quite good example is treatment of dry eye syndrome. And yeah, we have to be aware of that it is perhaps the most common eye disease, affecting at least 5% of the global population. The reason for dry eye syndrome is that the mucus layer coating your ocular surface is leaky, so there are gaps as illustrated here. And then this is highly irritating when this protective layer is missing. You can even stain these gaps, these holes in your mucus layer with dyes like rose Bengale. So here you see these gaps in the mucus layer that should cover the ocular surface, and that leads to this inflammation and irritations like shown right here. Based on that knowledge, we decided to use thiolated polymers to fill these gaps because they really mimic the natural mechanism. When you instill eye drops containing thiomers, thiomers form disulfide bonds with mucus glycoproteins, as already explained to you, and they also cross-link with each other. And in this way, you can really nicely fill these gaps on the ocular surface, and you see really a tremendous improvement in the symptoms. So those are data from clinical trials. These studies are done with a so-called fluorotron. This is one of my former PhD students who participated in one of these trials. And you use sodium fluorescein as marker. And as you can see here, before the treatment, the sodium fluorescein is not really properly evenly distributed over the ocular surface, which provides evidence that the mucus layer is not really intact. Whereas after treatment, you see an even distribution of the sodium fluorescein over the ocular surface. And here you see the data from another volunteer. Perhaps here the difference is even more impressive. These thiolated polymers for treatment of dry eye syndrome, they were then further developed to a product called Lacrimera, which is meanwhile on the global market. And here are data from late-stage clinical trials showing the improvement in the symptoms over numerous weeks of treatment. You find no other polymer on the global market that would show an improvement in the symptoms. So the thiolated polymers are the only ones that provide really an improvement in the symptoms in treatment of dry eye syndrome. And these data, they were obviously that convincing that this product won in 2018 the German Innovation Award. So it's quite challenging to get that award, and the company that developed the product won this German Innovation Award. The slogan for the product is "Tears for Dry Eyes." In my title, I mentioned also new developments, so at least I would like to share one newer development with you, and this goes in the direction of antiperspirants. Even in the sweat ducts, you find a mucus gel layer, which is shown right here. And antiperspirants that are currently on the market, they work in the way that they simply seal this, they plug this sweat ducts, and in this way, the sweating is reduced. Still gold standard is aluminum chloride, which is shown right here. The mucus layer has always an anionic charge because of sialic and sulfonic acid substructures. And when you use a cationic polymer like aluminum, it interacts ionically with the mucus, it forms a plaque, and in this way you can simply block the sweat production. But aluminum chloride is, we meanwhile know that it causes Alzheimer's disease, so the industry certainly tries to get rid of aluminum salts for that treatment, and a likely promising alternative are thiolated cyclodextrins, which is illustrated right here. The tests are quite, I would say, quite, even entertaining. You can use microfluidic techniques to mimic these sweat ducts, and you can then investigate whether you see such a plaque formation or not. The reason why aluminum is used and not other multivalent cations like iron 3, for instance, or zinc 2, is that aluminum forms actually much bigger clusters in the size range between 1 and 2 nanometers, and that is needed for this plaque formation. And thiolated cyclodextrins, they are also in this size range, so we are quite optimistic that we can sooner or later provide an alternative to aluminum salts to treat hyperhidrosis. Yes, that brings me to the next point, permeation enhancing properties of thiolated polymers. The mechanism for a tight junction opening, and that is one of the reasons why thiolated polymers are permeation enhancing. The mechanism is quite complex, so I won't go in detail on the mechanism. It took us quite a long time to discover this mechanism. In a few words, what is taking place is that thiolated polymers inhibit an enzyme on mucosal membranes that is responsible for the closing of tight junctions. So when this enzyme is inhibited, tight junctions remain opened. Almost 20 years later on, a Chinese research group went then again in detail on the mechanism behind. And on the one hand, they could confirm the mechanism we discovered, but on the other hand, they also showed that it is actually much more complex than we assumed, let's say, in 2002. But you can, of course, make use of this permeation enhancing tight junction opening effect. This is just one example. Here also focusing on insulin, as in the last talk. And here we could show a quite high oral bioavailability of insulin in rats by making use of this tight junction opening effect of a thiolated chitosan. We compared then also the permeation enhancing effect of a certain thiolated polymer with still the gold standard regarding permeation enhancers, namely medium chain fatty acids. We used here sodium caprate, and we could show in this study that the thiolated polymer shows an even higher permeation enhancing effect than the sodium caprate when we use it in exactly the same concentration. Apart from an improved permeation on the gastrointestinal mucosa. We see this effect also on the ocular mucosa. On the one hand, as already explained to you, we see this prolonged residence time. And on the other hand, we see also this permeation enhancing effect. That image is from the cover of a cyclodextrin journal. And the cells, how they are illustrated right here, they are not, I would say, this is not how corneal cells really look like, but still I like the image because it shows in a quite nice way how these systems are working. They adhere to the epithelium, and then by inhibiting certain enzymes, we see this opening of tight junctions and improved uptake. And this is also shown here via the results of permeation experiments. Thiolated cyclodextrins work also permeation enhancing on the gastrointestinal mucosa. And in this study, this is also an in vivo study, we could show a pronounced improvement in the oral bioavailability here of furosemide when you incorporate this drug in a thiolated cyclodextrin. Another mode of action when we stay with the thiolated cyclodextrins, and that's a quite recent result, is the uptake of thiolated cyclodextrins per se. This is shown here when you incorporate drugs, and here we use the marker, fluorescein D-low rate. When you incorporate that in a thiolated alpha cyclodextrin, you see more than, I will show you in the next slide, more than 20-fold improved uptake by here CACO2 cells, whereas when you use the unthiolated cyclodextrin, you see only a very minor uptake. So that will likely be of potential for the future, for instance, for anti-cancer drugs that are quite hydrophobic and that are immediately pumped out of target cells. So we might make use of the thiolated cyclodextrins for such applications. This is the slide I mentioned. Here you see this tremendous, more than 20-fold improved uptake of a marker compound of the fluorescein D-low rate by incorporating it in a thiolated alpha cyclodextrin. Furthermore, thiolated polymers inhibit also efflux pumps like P-glycoprotein. The reason for that is also quite simple. Within the channel forming unit of these efflux pumps, you find quite often free thiol groups, and they get, this is at least our working hypothesis, they get cross-linked by the formation of disulfide bonds. And here data from in vivo studies where we used the substrate for P-glycoprotein, and where we could show that by using the thiolated polymer, you can also tremendously improve the oral bioavailability of this drug. Furthermore, we very recently discovered also that thiolated cyclodextrins, they also show this efflux pump inhibitory properties. So this is a publication in preparation. That brings me to the next point, a sustained drug release that can be provided by thiolated polymers. On the one hand, we can provide such a sustained release by covalently attaching drugs that bear a thiol group, and many peptides like this example here bear at least one free thiol group. So you can attach these drugs to a thiolated polymer, like here a thiolated dextran. And by doing so, you can tremendously improve the elimination half-life of, in this case, the peptide drug. So here the elimination half-life of the peptide is really short. When you attach it to a thiolated polymer, you inject it intravenously, you see then a tremendously prolonged elimination half-life that is highly beneficial for numerous drugs when you can prolong this half-life. The release from the polymer is taking place by thiodisulfide exchange reactions with glutathione. You find also in low concentrations in the bloodstream. And another mode of release is shown here. You can incorporate drugs in thiolated polymers and you stabilize these drugs in this three-dimensional network by cross-linking your thiolated polymer. So the drug remains inside your, for instance, nanocarrier system, and after cellular uptake within the cytoplasm, as we find there in at least 100-fold higher concentration in glutathione, these disulfide bonds are reduced and your drug is released. So far, there are no product developments in the pipeline, but it's a quite interesting mechanism, and it is also based on thiolated polymers. This brings me already to the last point I would like to share with you within my talk today, namely the complexing properties of thiolated polymers. Another word for a thiol group is mercaptor, and that's a hint that thiol groups bind metal ions. It comes from Latin mercurius captans, mercaptor. So they bind metals, not just mercury. We know, for instance, that thiol groups are tightly bound to gold. So you find a lot of gold particles that are decorated with thiolated compounds in general, but also with thiolated polymers and thiolated cyclodextrins, making use of this mechanism. But products that are on the market, global market, using thiolated polymers, they are heading in another direction, and this is illustrated here. You find, for instance, products containing thiolated chitosan on the market to treat nickel allergies, because nickel is tightly bound to these thiolated polymers, and by binding the nickel, you can then decrease the allergy by simply removing the nickel from your skin. Yeah, having said that, I can already come to the conclusion of my talk. Within my introduction, I mentioned that the great potential of thiolated polymers for life sciences might be described in the best way by the simple imagination of protein chemistry with and without cysteine. Having shared with you all these results, I hope that you agree with me that there is really great potential in thiolated polymers, and we find more and more applications for them in the biomedical field. In particular, I would say because of their bioadhesive, in situ gelling, permeation enhancing, drug release controlling, and metal binding properties. They are already used for numerous biomedical applications and in numerous products, but I'm sure over the years we will find more and more further applications in the biomedical field. Having said that, I wish to thank all my co-workers, my whole team for their contributions. I showed their names always like here on the bottom of my slides when I referred on the publications. The group told me they prefer it in that way, not showing just a picture of the whole group. They regard it as more efficient, so I wish to thank them. Then, of course, I wish to thank Vitaly and Sergey for inviting me, and I wish to thank also you for your attention. Thank you very much.